The body operates on a strict mechanical equilibrium known as homeostasis. The central scale shows the balancing act between metabolic functions and regulatory systems. But when chronic stress permanently exceeds the body's capacity to regulate itself, this structural support fractures and the entire system collapses. Tracing the origin of almost all carcinomas, we find a unified foundation: impaired cellular energy metabolism, driven entirely by this cascade. This diagram illustrates those four failures. The continuous arrows surrounding the central core of cellular dysfunction show they form a self-reinforcing vicious cycle. They operate sequentially. The chemical exhaust produced by one failing system serves as the direct catalyst to ignite the next, locking the cellular environment into a state of continuous degradation. Our objective is to trace this precise biochemical pathway. We will follow a tumor's derivation from a single failure inside a cellular power plant, all the way to its ability to evade the host's immune system. Tracking this map, we see that cancer is a predictable, end-stage biological adaptation to these four cooperating mechanisms of stress. The cascade begins at the subcellular level with oxidative stress. Inside the mitochondria, extreme metabolic stress causes the cellular power plants to effectively rust from the inside out, leaking volatile molecules. This schematic details the failure. When the electron transport chain breaks, toxic reactive oxygen species escape. Hypoxia forces the production of nitric oxide, creating reactive nitrogen species. Among these, the hydroxyl radical is the most cytotoxic agent. It operates by physically stealing electrons from nearby structures. When it targets the nucleus, this oxidation process directly damages the DNA, initiating mutagenesis. The resulting mutated, oxygen-starved cell creates a permanent source of chemical irritation. That irritation acts as the strict biological prerequisite required to trigger the second pillar of the cascade. We usually understand inflammation as a healthy, temporary response to an infection. Chronic inflammation occurs when the immune system fails to terminate the healing process. This flowchart compares the pathways. Top track, acute inflammation resolves. Bottom track maps chronic irritation. Dr. Griffin describes this as the overhealing theory. The body endlessly tries to repair a site that is constantly re-injured. Because the damaged mitochondria are continuously leaking reactive oxygen species and other toxins, the local tissue is forced into a perpetual state of attempted repair, followed immediately by re-injury. Carcinoma exploits this permanent inflammatory state, using the continuous influx of immune resources to transition from localized cellular damage to uncontrolled growth. This entire process is accelerated by a systemic catalyst. This triangular diagram maps the vicious cycle of metabolic syndrome driven by insulin resistance. Chronically high blood glucose effectively jams the cellular locks. The cells stop responding to insulin, prompting the pancreas to flood the body with even more insulin, a potent growth hormone that fuels local inflammation. This molecular diagram illustrates glycation, the caramelization of internal tissue. Glucose cross-links with proteins, forming advanced glycation end products, or AGEs. These deformed proteins bind to specific RAGE receptors on the cell surface. Activating these receptors initiates the biochemical suppression of the p53 gene, which biologists refer to as the guardian of the genome. Normally, p53 forces a damaged cell to undergo apoptosis, or programmed suicide. By blocking the p53 gene, that safety mechanism is completely disabled. By using excess sugar to neutralize its own self-destruct sequence, the mutated cell achieves functional immortality. Now functionally immortal, the growing tumor faces an immediate logistical problem. Its exponential growth is physically constrained by a lack of incoming nutrients. To solve this, the tumor exploits the continuous, localized inflammatory response from Pillar 2 for tissue to release specific growth factors. This illustration demonstrates angiogenesis. The tumor hijacks the body's normal wound healing protocols to construct its own dedicated vascular network, sprouting a dense, erratic supply of new blood vessels directly from a healthy artery. With a dedicated supply line siphoning nutrients directly from the host, the final biological barrier to exponential growth and systemic metastases is removed. This brings us to the central biological paradox of neoplasia. Despite its aggressive growth, the tumor runs on a highly primitive, inefficient metabolic engine. This chart explains the engine. Stress triggers a fight-or-flight response, dumping sugar into the blood. The flooded cell undergoes the Warburg effect, abandoning its rested mitochondria to consume massive sugar without oxygen. This primitive process generates toxic lactic acid, severely dropping the localized pH. By actively engineering this acidic, hypoxic microenvironment, the tumor gains a distinct survival advantage, biochemically paralyzing approaching natural killer cells before they can attack. As Panel 4 illustrates, the body's primary defense mechanism is paralyzed and repelled by the acidic shield. Carcinoma functions through the active engineering of a sugar-rich, acidic, and oxygen-deprived fortress, meticulously designed to guarantee its own survival. Since the pathological cascade follows a predictable mechanical sequence, each stage presents a specific biochemical target for intervention. Halting the chemical fuel feeding the 4 pillars requires eliminating the inputs in this red column, specifically sugar, trans fats, and processed meats. Once the fuel is cut, targeted nutritional chemistry can be used to launch a chemical counteroffensive. Specific compounds act as precise biological tools. Glutathione directly neutralizes the oxidative stress in the mitochondria, while omega-3 fatty acids and certain phytochemicals biochemically target the tumor's hijacked vascular network. Specific nutritional inputs function as a chemical instruction set, capable of dismantling the tumor's supply lines by addressing the underlying biochemical drivers. Advanced metabolic protocols can directly counter the tumor's engineered environment. This chart displays 3 primary interventions, starting with ketosis. By restricting carbohydrates, the body switches its primary fuel source to ketones. Because the Warburg effect relies exclusively on glucose to function, this transition effectively starves the mutated cells while providing clean energy to the brain. To address extreme oxidative stress, molecular hydrogen selectively targets the highly toxic hydroxyl radical, safely neutralizing it into harmless water. Next, neutralizing the tumor's lactic acid shield requires restoring systemic alkalinity. Calcium helps restore a balanced pH to the surrounding tissue. When that acidic barrier is cleared, the biochemical suppression on the p53 gene is lifted, and the reactivated natural killer cells can begin clearing the mutated tissue. This final diagram illustrates the endpoint. Identifying and removing the pathological stressors feeding the 4 pillars allows the body's natural drive for homeostasis to restore the architecture of health.